Immune regulation and tolerance, another topic in immunology. Now we are moving to immunopathology. This is the basic of immune regulation related to autoimmune response. First of all, we would know what is the basic. The that is the immunological equilibrium. That is, there is a balance always between lymphocyte activation and control. So this balance, now it's in a balanced state. There is activation of effector T cells by means of immune response. At the same time, there is a tolerance, that is capability of immune. One way we'll say tolerance. The way they are coping up these activated cells activity. That is termed tolerance. It's done by means of regulatory T cells. So in a normal response, following activation, reaction against pathogens like inflammatory disease, reaction against cell. That's a disease. Other way, they react against those pathogens. But ultimately, this overactivation leads to disease for cell, that is autoimmunity. But with the presence of tolerance, regulatory T cells, this overactivation one way, activation will be controlled, tolerated, regulated. That's done by these tolerated or regulatory T cells. Control response to pathogen, no response to cell. That's how it goes with the presence of this balance. How it occurs? For that, there are several mechanisms. We would divide them as central and peripheral. Central by means they generative lymphoid organs that are those are thymus and bone marrow. We know those are the primary lymphoid organs. So here there is a tolerance mechanism for cell. Once lymphoid precursor produce immature lymphocytes, there is a recognition of self antigens in the thymus. If these lymphocytes are capable of identify self or own body antigens, that is danger. It is disastrous. To prevent that, there is a check, policeman-like activity by thymus, other cellular T cells and other lymphoid cells does. So from that, if those immature lymphocytes who are capable in identifying self-antigens are going to be destroyed in several mechanisms. One may they trigger apoptosis, leading to deletion. Other change in receptors, receptor editing B cells. Other way is development of regulatory T lymphocytes. They form regulatory T lymphocytes, CD4 plus T cells only. So once these cells are liberated into the periphery, peripheral tolerance is there. That is in the peripheral tissues. Same way, there are these mature lymphocytes now. They are again, if they are capable of identifying self-antigens, this is the site where in the periphery they act on various tissues. They trigger energy and apoptosis trigger, or there is a way of suppression by means of the presence of T regulatory lymphocytes. That's how it happens. So about the self-recognition, antigen recognition in the thymus, still negative selection or deletion. Now this is immature T cells, this antigen present in cell, probably dendritic cells. Immature T cells specific for self-antigens are there. So later on, with the presence of these, they produce regulatory T cells. And other one, they destroy this self-reactive lymphocyte. And now they liberated into peripheral circulation as regulatory T cells. That's how it goes. Negative selection leads to deletion and development of T regulatory cells. That's how. Same thing what I discussed here is there. Negative selection and development of regulatory T lymphocytes. What self-antigens are seen in the thymus? ubiquitous cell-associated and circulating proteins. Every antigen is there because it's part of our body circulatory organ. The thymus has a special mechanism for displaying peripheral tissue antigens in thymus medullary epithelial cells. So that's how it displays, where they signal self-reactive lymphocytes for death. That's how it goes. They produce when then those T lymphocytes in immature comes. If they identify these signals, then they trigger apoptosis, deletion, and leads to destruction. And some maybe go for the formation of regulatory cells. That's how it goes. So about the peripheral T cells tolerance. In a normal response, with the presence of dendritic cells, with the antigen presentation and co-stimulator signal, B7, CD28, and T cell receptor, these two co-stimulator signal, major and co-stimulator signal should be there to form activated T cells and memory cells. It's a normal mechanism. But with the presence of these, if these cells are capable in identify self-antigens, as said earlier, they trigger energy. There means cell is there, no destruction, but functional unresponsiveness is there. Other one is other way is suppression. It's by the presence of T regulatory cells, what I said earlier, blocking activation. Other one is they trigger apoptosis and leads to deletion. What is T cell energy? Bit more. Here it's antigen recognition, but other thing is no co-stimulator signal. So there is no such activation. It's unresponsive. Other way is ligand is there, but it binds to inhibitory receptor rather being CD20. So again, no co-stimulator signal is there. So signal block, no activation. So it is unresponsive. So one way. Other way is same thing, CTLA4. Just to know, with the antigen presentation, co-stimulator signal, here that same thing, inhibitory cell. Sorry, inhibitory receptor. So no, there is no such activation. So that's what happened in the chronic infection, chronic viral infections like hepatitis B, C, HIV, HLTV, or even herpes viruses. Likewise, there are chronic infection rate is there. So in this case, in the usual ways, naive CD8 or cytotoxic T cells, in the presence of virus, it's further activated and go for memory T cells, enhance antiviral response. In acute infection leads to clearance of the virus. But in a chronic infection, there is a persistence of virus. Why? It's exhausted T cells, unable to respond to virus. That is their energy. Expression of inhibitory receptors like here, CTLA4. Now, they can't activate it. They cannot activate. That's what it's doing. So that's a basic mechanism. So once you know these, in the next one, we can discuss about the autoimmunity. In addition to that, there are some therapeutic potentials of introduction of regular T lymphocytes into body. It's mainly used for graft rejection. There are clinical trials. One for liver transplantation, signal infusion of T regulatory cells resulting in tolerance. So they are rare. It's on the way. Like, thank you.